So you're sitting at home, you're reading the news, and you're thinking to yourself, man, I'm glad this whole COVID-19 pandemic is over. But then you start seeing articles from the Wall Street Journal, CNN, MSNBC, all saying the lab leak most likely origin of COVID-19 pandemic, according to the Energy Department. And you ask yourself, well, hold on a second. I thought the whole thing originally was that COVID-19 didn't come from a lab. I thought the whole thing originally was that was a conspiracy and that was taking away from the actual problem at hand. But now the Energy Department is saying that COVID-19 came from a lab. And you also ask yourself, and what business does the Energy Department have making assumptions that pertain to virology? Well, because you're an avid listener of the science behind that, you shoot Atticus at the science behind that an email. So let's talk about the science behind that.
Welcome to the Science Behind That with Atticus Hamilton.
Hello all you scientists, and welcome on back to the Science Behind That podcast. I hope you guys are having a good Monday. I know I am. This episode is going to be a lot of fun. Especially because for this episode, it was less of me having to do research into the topic itself, and more of doing research into the conspiracy theories that are out there. And, that's always fun. We always like reading conspiracy theories, just for fun, I guess. I don't know. It was very fun to do this. And so I do have a couple things here to say before I get started. First off, I'd like to thank the listener who sent me the email recommending this topic. This was a great idea, and frankly, I should have come up with it sooner, But yeah, thank you for that. This is a great idea. I hope you enjoyed today's episode. And the other thing I want to add here, look, this is not a politically motivated episode topic. But what is motivating me for this is, just a wee bit of anger. Okay, a wee bit of anger in the sense that I thought this was done. I thought this idea was put to sleep. Because really, in all honesty, there's no evidence, at least that I've seen, that really supports this theory. And I'll even dive into the, I live in America, so I'll dive into the intelligence community officials, air quotes, evidence that COVID-19 came from a lab. And this is from the White House Oversight Committee. This was during a press release that was held on the SARS-CoV-2 pandemic. So we're going to talk about that here. There's a ton of stuff in here, like the questions that cannot be solved by science alone. Pretty much everything can be solved by science, even if we don't have the technology for it yet. Theoretically, anything could be solved by science, because science is the lens at which we look through the world, or the lens at which we look through to see the world and to solve problems, right? So science isn't it's not a thing, it's just a way of solving problems. But anyway, that's just one of the many problems with this. But we're going to get into it. So this episode is going to be fun. There's not that much biochemistry that's going to be going in here. I'm going to, summarize it for you nicely, I think. So grab yourselves a nice hot cup of coffee, add a little bit of maple syrup to it, and without further ado, let's jump into today's episode of The Science Behind That. All right, so where to begin? So, when doing this episode, or I guess when doing the research for this episode, I found a lot of interesting things and a lot of interesting justifications for why people were, or why the lab leak theory is the most likely theory. And hopefully we can, I guess, debunk it. Or at the very least, just provide you the evidence and the science behind it so you can make your own informed decisions. So, okay, so, where to begin? So I think the natural part of this, of where to begin, is let's first talk about how you would theoretically bioengineer a virus to, be super infectious, right? And so this is part of gain-of-function research. And so gain-of-function research is what was being done at the Wuhan Institute of Virology. And gain-of-function research is often used a lot as sort of a buzz phrase in media because there's a little bit of misinformation about it, right? So people say, well, gain-of-function research looks into developing bioweapons. So therefore, because they worked with coronaviruses at the Wuhan Institute of Virology and they were doing a broad type of research that contains research looking at how to develop bioweapons, COVID came from a lab. So you see, there's a lot of just correlation there. So gain-of-function research doesn't just pertain to bioweapons, but in general, gain-of-function looks at how organizations or how people can take something that's already present and manipulate it to fulfill a certain task. A lot of times it ends up being bioweapons, right? And so, in the 1980s, Russia was doing a lot of, air quotes, gain-of-function research on smallpox, and it was discovered that their gain-of-function research was actually just manufacturing smallpox bioweapon stockpiles, right? And that was in the 1980s. That was about 3 years after smallpox was eradicated from the earth. And for those of you who don't know, smallpox is a select agent in America, which means it is an agent that is highly regulated because it can be used for bioweapons, and it would be a very effective bioweapon depending on how you define effective. And it's a Category A select agent, which means, so you have like Category A, B, and C, and Category A are the I guess, best select agents. It's a weird way of phrasing it, but they're the most risky, I guess, in terms of the most likely for an enemy to use in bioweapon work. So, so that's generally speaking what gain-of-function research is. I've done gain-of-function research before, or at least I've had a little bit of exposure to it. It's not as hyped up. It's not all as hyped up as everybody seems to make it or say it is. But anyway, so that's gain-of-function research. So let's talk about how you would actually take an organism, right, and bioengineer it into a bioweapon form. And I'm not saying this to, give any of you ideas. Please don't do that. I'm talking about this because it's really important for us to understand where these conspiracy theories fall short. It really is, at the end of the day. It is very important. So, how would you do it? Well, first you need a scaffold organism. And what I mean by that is, we do not possess the technology to design viruses from the ground up 100% from scratch. Now, some of you may say, well, Atticus, researchers created the genome for horsepox from scratch, and, horsepox is extinct. Well, they didn't create it from scratch, right? The genome for horsepox was already known. So if you know the genome for a virus, then it's really easy, of course, right? So if you have the entire genetic sequence of horsepox, for example, it is really easy to get custom DNA or RNA made. It is so easy. And there, a couple years ago, I think it was like 2017, there were researchers in Canada who were doing gain of function research. But the research specifically was looking at how easy would it be for a bioterrorist to take a genetic code which is publicly accessible, like the genetic code for smallpox is publicly accessible, the full genome for horsepox is publicly accessible. And they were looking how easy would it be for somebody to take that publicly accessible genome and submit that to one of the numerous companies out there that manufacture custom DNA or RNA for people, and then would that then be infectious? The answer to all of this is very easy, and yes, it would be. It would be infectious because all you'd have to do is what the researchers found is once they got this genetic material, all they needed to do was infect a cell with a syringe with the genetic material, and then, bing, bang, boom, the cell does the rest. And then they were able to actually produce horsepox, which had been extinct, in the lab, which was pretty scary. And since then, a lot of these companies that design custom genomes for people have they've put in safety measures, right? They have algorithms that check for certain conserved sequences of the genome to see if it actually is, if somebody actually is putting in there the full genome of, smallpox or horsepox. And why is this possible? Because the genomes of these viruses are relatively small. Humans have like a 3 billion base pair long genome, but viruses, some of them have like 2,000 base pairs. So, I mean, it's really tiny. And so that's what you'd have to do first, right? And so you need a scaffold organism. So whether or not that's taking something that was previously in existence and just making the genome from it, from the genome that already exists, or you take an organism that already exists and you alter it a bit. And so this is the most common conspiracy theory I see for SARS-CoV-2. And you may notice I'm calling it a conspiracy theory as opposed to a theory because there's a ton of evidence in opposition to it, right? And so in science, for something to be considered a theory, there has to be all the evidence in support of it, but no evidence in against it, in opposition to it. But this is different. It has evidence, I guess, in support of it, and it has some evidence, or a lot of evidence, against it, right? So it can't be a theory. At best, it's a hypothesis. So I'll call it a hypothesis. But anyway, so the most common hypothesis I've seen for the lab leak idea is that the Wuhan Institute of Virology took a already existing coronavirus. I think the one that I saw most commonly was SARS-CoV-1, which was the virus that caused the SARS epidemic in the early 2000s. I think it was, 2003, in China. It sickened like 800 people. It sickened like 800 people. It was pretty well contained because the virus kind of sucked at spreading person to person. But anyway, so the theory then would be you take that organism and then you modify the genome to make it more infectious. So the question is, how would you modify it, right? Because again, we can't build organisms from the ground up. And to that point, we also can't build genetic traits from nothing, unfortunately. That would be cool if we could. I mean, if you could just be like, hey, I want a pig that flies, and you just build a genome or a gene that encodes for creepy pig wings, and you insert that into a pig as a plasmid, that'd be so cool, right? Get flying pigs, but, can't do that. So you need to take a gene that's already present in some other organism. Generally, that has to be compatible, kind of like writing code. But it becomes different than writing code because it's almost as if It's more of like it's more of like finding a code that operates on the right operating system, right? So if you're encoding a program that'll only work on Linux, you can't put that program on a Windows operating system. It won't work. And what I mean by this is, in microbiology and virology, you have to be very careful with how you insert genes into genomes. Generally, it's done on plasmids. There are other examples where you can use a CRISPR-Cas system to remove the gene that you want to remove and then insert another gene sequence in that place. So you can do that. It's more time consuming. And it's not as clean as plasmids are. But anyway, you need to be very careful. So let me give you an example. So in the real world, I don't know if any of you have ever asked this question, but where does insulin come from? Well, insulin, we no longer produce it from pigs anymore. At least pharmaceutical grade insulin, we don't get from pigs anymore. We get from yeast. So yeast are one of the very few eukaryotic organisms, which are organisms with a nuclei, that can accept plasmids. So it's actually pretty cool, because what researchers were able to do is they could take a yeast plasmid, and a plasmid is basically just a circular piece of genetic material that you can insert genes onto. So it's basically like, I don't know, it's like those little robots that you see carrying around packages to people's doors, right? So it's just a little circular piece of genetic material that you can add a gene onto. Now, the gene can be pretty much from anything, really, as long as the genomes are compatible. And what I mean by that is, you can't take a gene that's all encoded in RNA and put that in a DNA plasmid. Although, even then, you kind of can if you convert that RNA gene to DNA. But then, okay, anyway, that's too in-depth for what we really need. So, for insulin, they took the gene encoding insulin production in pancreatic cells of pigs. They inserted it into a yeast plasmid, and then they inserted the plasmid into the yeast cells, and the yeast then started transcribing that plasmid and translating that plasmid. And so now we get yeast in the laboratory that produce a ton of insulin. And then that insulin is used in pharmaceutical products like for diabetes. So that's what I mean by you need a scaffold, right? They didn't just design an entity from the ground up. They used a yeast cell, then they used a yeast plasmid, and then they took a gene from a pig, put it in that yeast plasmid, and put that plasmid in the cell. Very similar with viruses, kind of, except with viruses, you're not really going to use plasmids. And the reason for this is because viruses don't transcribe their own genomes, right? They don't really do any converting the DNA to RNA, or they don't do any reverse transcriptase, which is converting RNA to DNA, and they don't do any translation. They don't make proteins. The viruses don't. That's why they infect the cell. The cell does that all for them. So how do you alter a virus's genome? Well, depending on the structure of the virus, if it's enveloped, you'll remove the envelope, right? You'll lyse the envelope, which is really easy because envelopes are really delicate. And envelopes are it's like a bubble of phospholipids, or the cell wall. You could think of it like that. It's a bubble of cell wall that surrounds a capsid, and the capsid of the virus is going to be protein. And then the protein, you can just use a protease to break down that capsid, and so then you have the free genome. And so then with viruses, you're going to use a CRISPR-Cas system. Now, there are a lot of different CRISPR-Cas systems depending on what you're editing. So CRISPR-Cas is really good for DNA, but there are others that work better with RNA. So ultimately, how does this relate back to COVID? Well, if the Wuhan Institute of Virology was making a bioweapon, and if they were going to release that on the world, on their own people and destroy their own economy, well, how would they do it? So they would probably take, the conspiracy theory I saw is they took the original SARS-CoV-1, then they used a CRISPR-Cas system, and they would have to remove from SARS-CoV-1, they would have to remove one of many potential things. The problem with that one was it didn't really bind very well to human receptor cells or to human receptors, right? And that was the problem. So coronaviruses have a variety of different receptors they can bind to, and it's pretty interchangeable because in nature you'll find some coronaviruses that only bind to ACE1, and you'll find others that only bind to ACE2, and then there are a variety of other different receptors that they can bind to. And so what they would have to do is they would have to remove the receptors that the original SARS bound to, because it just didn't do it very well. And so it took a long time to sort of enter the cells and replicate. And they would have to change it, because the other thing about the original SARS was the original SARS bound to receptors that were more commonly found in the lower respiratory tract, right? And very few other places. And so it presented a problem when you look at spread for the virus. So the original SARS-CoV-1, then, it couldn't spread as well as SARS-CoV-2, right? So it required much closer contact to spread, and that's one of the reasons why it sucked at spreading compared to SARS-CoV-2, where it can spread asymptomatically and it's aerosolized. And that just gets down to where the receptors are. We have ACE2 receptors all throughout really our respiratory systems. So there are ACE2 receptors in our throat, in our mouth, in our upper respiratory tract, in our lower respiratory tract. And that really allows the virus to perfuse pretty much everywhere along the respiration pathway. Of course, there's more evidence too that shows that it can spread in other bodily fluids. But the main route of transmission for SARS-CoV-2 is respiratory. So if they really were creating a bioweapon, which is SARS-CoV-2, if they really were creating it from SARS-CoV-1, they would have had to take in a different receptor gene. They would have had to remove the original receptor gene from SARS-CoV-1 and take in a receptor gene from another coronavirus that maybe has an ACE2 receptor, and then inserted that. And that gets into the second point here, which is a similarity, right? It's a similarity. And what I mean by that is we possess the genetic technology where we can see how similar different organisms' genomes are to one another. And that's part of how we develop phylogenetic trees, right? Or trees of relations. Sorry, I needed a coffee there. So I actually looked this up because I wanted to give you a very specific number. So how similar is SARS-CoV-2 to SARS-CoV, or the original SARS? 86.85%. Now, why is that significant? It's significant because if it was bioengineered and they only changed one gene, it would be more like 98% similar. It wouldn't be 86.85% similar. And that, to me, the fact that it's 86.85% similar, to me, suggests one big possibility of where SARS-CoV-2 came from. Animals. SARS-CoV, the original one, right? It disappeared after 2003, and we never saw it again, right? It disappeared. To put a personality onto it, I think it just retreated into an animal reservoir, into a host reservoir. We don't know what that reservoir is. Now, there are a lot of coronaviruses that come from bats, but there's also coronaviruses that come from pangolins and snakes and a whole different host of animals. So we don't know what that reservoir is, but what we do know is this. Sometimes a cell can be infected with 2 viruses at once. And when that happens, genomes get shuffled, basically. And so in that case, so you have antigenic shift, right? And antigenic drift. And those are just 2 different forms of mutation. That's not referring to when 2 viruses infect a cell at the same time, but that's referring to the severity of, or the significance of a mutation. But when 2 viruses infect a cell at one time, the result is going to be usually a whole different virus, because all those genomes, assuming that they're similar viruses in terms of replication mechanisms, those genomes are going to be switched up entirely. And so that's most likely what happened here, is the original SARS coronavirus was just circulating in a population of reservoir hosts in China. Because, I'm sorry, but a lot of diseases come out of China, because there's a lot of animals in China, and there's a lot of different climate conditions in China, and a ton of migratory birds that circulate the entire globe go to China. So it's just a perfect storm, right? Then the other theory I saw is that the Wuhan Viral Institute of Virology was working on a bat coronavirus called RATG13, and that had a 96.75% similarity to SARS-CoV-2. And, when I saw this, there were a lot of people that were like, oh, that's it. That's exactly where it came from. No, that's not where it came from. RATG13, it may be 96.75% similar, but it can't infect humans. It can't infect humans. So that 3-ish% that's different is probably what allows SARS-CoV-2 to infect humans. And with this, it's not a matter of, you just remove a receptor. No, because the entire replication cycle is different for RATG13. So again, let's recap, shall we? Ultimately, to create, I guess, a bioweapon, as the conspiracy theories allege, to create a bioweapon, you need a scaffold organism, right? You need something to base it off of. And then B, you need to insert genes or selectively remove genes from the organism you're looking at to allow it to fulfill whatever task you want. Now, you can't make it magically only infect gorillas. Well, actually, you might be able to. I don't know. It depends on if there's cell receptors that are unique amongst gorillas. Maybe there are. I don't know. But you can modify an organism to do certain tasks to a point. But anyway, and if that were the case, you'd be looking at a genetic similarity of probably around 97% to 98%. I mean, it would be pretty close, especially with the most likely route of how you would design the SARS-CoV-2 virus. If you only had the original SARS, you'd be looking at much higher of a relation than what was it, 86.85%. So I mean, and I'm not the only one that says this. There is research out there that shares this same idea that demonstrates this. And as for the topic of the virus that was RATG13 that has a 96.75% similarity, other sources say that it's actually only 87% similar, right? 87% of the similarity is explained by RNA modification system in hosts. And what I mean by that is coronaviruses are RNA viruses, and so they have no proof checking mechanism. So natural mistakes happen. Mutations happen. I'm sorry. And as humans, it's hard for us to understand this because we have great proofreading mechanisms. But viruses, at least coronaviruses that are RNA, don't really. And so they found that 87% of the similarities between SARS-CoV-2 and RATG13 are by the RNA modification system and hosts, not from people creating a bioweapon, right? And then the other 65% are contributed by deaminations on cytidines or cytosine to thiamine mismatches. So they're ultimately there's no real good evidence that suggests, there's no research evidence that suggests that SARS-CoV-2 was created in a lab. And I hope at the end of this episode you at least understand why I'm saying that. Now this is kind of where I want to finish. So this goes back, now I'm going to go back to why the Department of Energy is saying that, oh no, it definitely came from a lab. So, ultimately, what they're saying is, so they started by saying, we're not here to, relay opinions. We're here to just relay facts. Although Dr. Redfield here definitely adds in his fair share of opinions. He literally says here, my informed assessment, which is an opinion. So the biggest piece of evidence, I guess, here that they have to say that SARS-CoV-2 came from a lab is that there were people who were sick at the Wuhan lab. I'm not kidding you. U.S. government information about a cluster of illnesses in that lab, which is exactly what you would expect to happen if the origin of COVID came from a laboratory accident, where a worker became ill knowingly or not and then took the virus out of the community. Okay, let us all take a minute here and realize how silly that sounds. So there's no actual data of blood serum analysis of those patients that show that they were infected with a coronavirus. So they could have been infected with a cold caused by a rhinovirus for all we know. But because they were sick in a lab that works with coronaviruses, according to the Department of Energy, that right there is enough evidence to suggest that SARS-CoV-2 came from a lab. Case closed. Additionally, former director Ratcliffe said that my informed assessment as a person with as much or more access to anyone to our government's intelligence during the initial year of the virus outbreak and pandemic has been and concludes to be that a lab leak is the only explanation credibly supported by our intelligence, by our science, and by common sense. Opinion. From a view inside the IC, if our intelligence and evidence supporting a lab leak theory was placed side by side with our intelligence and evidence pointing to a naturally occurring spillover, the lab leak side of the ledger would be long and overwhelming, while the spillover side would be empty. Speculation. Okay, so this is the problem I have, and this is why I did this episode. This is why that person who emailed me and said, Atticus, do this episode. Thank you. Thank you for doing that. Because this is what bothers me. There's no data here. They presented 0 actual pieces of analytical data that lend support to their hypothesis. Yet, they are making these statements as if it's truth. This is the thing, all right? He says, my informed assessment as a person with more access than anyone to our government's intelligence. If the evidence here really was irrefutable, there'd be 3 things happening. Number 1, the scientific community from the get-go would have already said, yeah, this is a contender of a theory. Number 1. Number 2, the genomes, there would be a virus there, a coronavirus there, that would have a 90+% similarity to SARS-CoV-2 that infects people. So not RATG13, right? And number 3, they would share it, right? They would share it, because if it really was a matter of public health, the air quotes intelligence community, if they had this amounting amount of data that showed that it came from a lab, they would share it. Or maybe they wouldn't. Who knows? They just make assertions without any evidence. And then they go on to say that their other evidence that it came from a lab is that China isn't giving up evidence about really what they worked with at the lab. And to me, that is just a phenomenally stupid argument. I work at a place that, if a foreign power was like, what do you do there? We wouldn't tell them. And there are a lot of labs in America where that is the case. There are a lot of labs in the world where that is the case. You don't just voluntarily give up the data that you're working with to another country, let alone a hostile country. So, to China, the U.S. is a hostile country. And to us, China is a hostile country, right? And, furthermore, China is a lot of things, but why would they release a bioweapon on their own people that would go on to literally eviscerate their economy with their whole getting to zero COVID policy? It eviscerated their economy. It doesn't make any sense. So, there you go. And of course, former director Ratcliffe highlights that anyone who disagrees with his theory that it was from a lab leak is politicizing it. So, that again, he's not sharing his opinion. So, ultimately, at the end of the day, that's why at least that is what I could find on why the intelligence community, Department of Energy, is now saying, yeah, it probably came from a lab, is just because of this White Oversight Committee press briefing where, in an amazing state of affairs for 2 days straight, everybody seemed to say nothing at all. So that was amazing. And in another fit of irony, another government service, the National Center for Biotechnology and Information, has said the COVID-19 lab leak theory is a tale of weaponized uncertainty. Scientists almost never say they're sure, and it could take years to pin down the pandemic's origins. Until then, people are trying to scare you. And, I don't disagree with that necessarily. Ultimately, we are pretty certain in the scientific community that it didn't come from a lab, for all of the reasons I outlined in the very beginning. And I invite you, I have a lot of listeners out there from Denmark, which is pretty awesome. So yeah, welcome. But I invite all of my international listeners, if you have any data as well that, leans one way or another on this, send them to me at thesciencebt@gmail.com. I'd love to take a look at it, and I think that'd be pretty cool. So, ladies and gentlemen, I really hope you enjoyed today's episode. Have a great week. I will see you on Friday. And, I always say this, and I think now more than ever, it is so important. Stand up and question everything.